Wow, he's huge. He must have been lifting for a while. I wonder how I've never seen him before. Hey, are you new here? Uh, how long have you been lifting for? I've lived here my whole life. Only been lifting for 2 months though. Only 2 months? I've been lifting for 2 years and he's way bigger than me and stronger than me. And the winner of day one, Big E Cheese. There's no way he's that big. After only 2 months of lifting, he has to be cheating somehow. Maybe he's on steroids. Maybe what he's using to cheat is in his locker. Hmm, there's nothing in here.
Excuse me, what are you doing in Big's locker?
Nothing. I was just looking for steroids. He's been only lifting for 2 months. There's no way he's not cheating.
Listen, I'm Big E Cheese's dad. You can call me Chuck. Take a walk with me and I'll explain everything.
So tell me, what does Big E Cheese have that makes him so muscular?
You see, Big has a very rare genetic mutation that affects his body's regulation of muscle growth. It's called myostatin-related muscle hypertrophy, also known as the double muscle mass mutation. This mutation affects the myostatin protein, which is responsible for regulating skeletal muscle growth. I devoted my life to myology, the study of muscles, after Big was born. This is where I do all my research.
That's amazing. So wait, what's a mutation and what causes them?
A mutation is a permanent alteration of the DNA sequence that makes up a gene, which usually causes some change in the phenotype, or physical features of an organism. There are many different types of mutations. Big's is something called a nonsense mutation. A nonsense mutation is a type of DNA mutation where a change in one nucleotide turns a normal codon into a stop codon. This causes protein production to stop too early during translation, creating a shortened and usually non-functional protein. For example, if a codon that normally codes for an amino acid mutates into UAA, UAG, or UGA, which are all stop codons, the ribosome stops building the protein before it is finished. Because the protein is incomplete, it often cannot perform its normal job in the cell.
So how does that one mutated gene result in such a big change in his physical features?
Well, it starts with RNA transcription. In this process, the DNA inside the nucleus of the cell is unwound, and the bonds are separated by an enzyme called helicase, as you can see in this poster. Then, the other enzyme used in the process, RNA polymerase, builds a strand of RNA by linking together complementary bases of RNA nucleotides.
What is a complementary base?
Complementary base pairing is necessary so the information stored within the DNA is properly transferred to the RNA so the correct amino acid sequence for the protein is created. The little letters on the poster represent the different nitrogenous bases on the nucleotide. For example, you can see that A, adenine, on the DNA strand pairs with U, uracil, on the mRNA because they are complementary bases.
Why is RNA used? Why can't DNA just code for the amino acid itself?
The main reason is to protect the DNA. In order for myostatin to be constructed, the blueprint for it needs to be brought into the ribosome so that it can be constructed. The RNA is a temporary copy of the DNA, so it can leave the nucleus, transfer its information, and then be deconstructed and recycled.
So what happens after transcription? How does the RNA create the protein?
That's where translation comes in. First, the mRNA from transcription carries the genetic instructions to the ribosome. The ribosome is made up of an RNA, which holds the tRNA and mRNA in its place and acts as a catalyst for the peptide bonds. tRNA, which has the anticodons for mRNA, brings the specific amino acids as shown on the mRNA. As you can see on the poster, the mRNA codon, AUG, is paired with the methionine amino acid and the UAC anticodon.
What's an anticodon?
An anticodon is the codon on tRNA that pairs with the ones on mRNA. It's similar to how a magnet is only attracted to the opposite pole. This system ensures that the proper amino acids, which are the building blocks of protein, are added.
So, how does this whole process lead to a constructed protein?
The ribosome first attaches to the mRNA, then reads the codon. Then, tRNA brings the corresponding amino acid and bonds with its anticodon. Once there are multiple amino acids, they will form a peptide bond catalyzed by the ribosome that links them together. Then, when a stop codon is reached, the protein is released. This is why an early stop codon affects the muscle mass so much, because the protein chain will be released prematurely and the protein will be non-functional.
Wow, that's a lot, but I think I got it. Thanks for taking the time to teach me. So, it turns out Big E Cheese wasn't actually cheating. He was just born with superior genetics that allow him to build muscle quicker and easier. Exactly. Hey, I just wanted to let you know that I'm withdrawing from the competition because I realized I had an unfair advantage and I feel bad. Loser! Get out of here! What happened? He just quit because he had an unfair advantage. I'm the winner for day 2 of the competition. Jerry! Oh man, he cheated.
Actually,